Hi everyone, I'm Elena, a third-year PhD student in Frank Aylward's lab in the bio department. Thank you so much for coming to my presentation, Too Big to See, Large Viruses Are Overlooked Players in the Ocean's Nutrient Cycles. Now, some of you may be wondering, what do viruses have to do with nutrient cycles? I thought they just caused pandemics. And yes, some viruses cause pandemics, but viruses can infect any living cell, and most viruses in the sea infect microbes like bacteria and protists. And when they infect these microbes, they release their cells, nutrients, or organic material into the ocean. And this process, where virus infections release nutrients into the sea, is called the viral shunt. And it can impact the concentration of nutrients in the ocean around the world. Importantly, it can impact how much carbon sinks to the deep sea. And this has important implications for climate change. So here's a picture of the deep sea. And these tiny little dots are globs of organic matter that include carbon called marine snow. And when this marine snow sinks to the bottom of the sea, it can get buried, which reduces the amount of carbon that can get released into the atmosphere. So viruses have important implications for climate change. But more broadly, viruses, because they infect all types of cells, can impact global change in the sea in many ways. They can cause infectious diseases, or they can reduce pollution by terminating algae that form blooms, or algae that release toxins. And as I mentioned, viruses impact nutrient cycles in the ocean, which impacts the overall health of food webs in the ocean and has implications for climate change. And my work is mostly on how viruses impact nutrient cycles, so I'm going to walk you through how nutrients move in the ocean to better illustrate where viruses come into play. So here's a picture of our ocean, and to start we have our classic food chain with algae that are photosynthetic and fix carbon. And these algae, which are not to scale, get eaten by smaller animals that get eaten by bigger animals, and so on. And all of these organisms, when they die or excrete waste, they release nutrients into the ocean. And these nutrients either sink to the bottom of the sea in the carbon pump, such as the marine snow you saw earlier, or they get eaten by consumer bacteria in a process called the microbial loop. And these relationships and processes in the ocean have been known for decades. However, only recently have we begun to understand how viruses affect these dynamics. Because viruses can infect all types of cells, they can impact these food webs at all levels. In particular, viruses can short-circuit how carbon moves up in food chains by infecting algae and releasing their carbon. In fact, viruses infect up to 40% of bacteria and 10% of protists each day in the ocean in some marine environments. However, the way we study viruses in the ocean has overlooked a major group of viruses. And the way we study viruses has largely been influenced by how viruses were discovered. Viruses were discovered by being able to fit through filters that bacteria could not. And so when we take our seawater samples and we want to enrich for viruses, we remove the bacteria by using these filters that are 0.22 micron in pore size, because we know that most bacteria are bigger than this 0.22 micron size. And once we enrich for our viruses, we can then start to study them. And we do this by several ways, such as sequencing their DNA in metagenomes, which are the DNA sequences of all organisms in a sample, or we grow them in the lab, or we look at them under microscopes. However, recently it's been found that viruses can be much larger than we thought. In fact, some of these viruses can be bigger than that 0.22 micron size. And one group of these large viruses is called jumbophages. Jumbophages are viruses that infect bacteria, and jumbos are defined by having genomes over 200,000 base pairs in length. For reference, the coronavirus genome is 30,000 base pairs in length, and a typical phage genome is 50,000 base pairs in length. So jumbophages are about 4 times the size of a typical phage. And I've shown that here with a picture of a jumbophage next to the smallest known bacteria, which is Mycoplasma genitalium, which is next to your typical phage. So jumbophages can be bigger than bacteria. However, they're thought to be rare in the environment. And we wanted to know, are jumbos actually rare, or do we think they're rare because we exclude them with these filters? So this led to our research question, how much are we overlooking jumbos by using a 0.22 micron filter cutoff? And next we wanted to know, do these jumbo phages matter? How much are they affecting the ocean's nutrient cycles? So to answer these questions, we started with metagenomes from samples collected everywhere. So again, metagenomes are DNA sequences of all organisms in a sample. And we wanted to get as many jumbo sequences as we could, so we looked at samples from elephant poop to New York subway surfaces to wastewater. We created a bioinformatic pipeline to detect jumbophage sequences in these metagenomes, and we ended up with 85 bait jumbo sequences. And we looked at the presence of these jumbos in seawater metagenomes. And we used this Terra Oceans data set because Terra Oceans has gone around the world collecting seawater samples and has filtered this seawater at different size fractions to enrich for different types of organisms. And some of these filters included those below that 0.22 micron cutoff and above that 0.22 micron cutoff. So this was the perfect data set to look at the presence of jumbos in samples that use different filter sizes, and also look at how jumbos might be affecting the ocean based on where they're found in the ocean. So to start, we looked at what samples our jumbos could be found in. And we found that most of the jumbos could be detected in samples both below that 0.22 cutoff and above that 0.22 cutoff. However, 21% of our jumbos could only be found in samples above that 0.22 cutoff. So already we saw that jumbos were being excluded by that 0.22 micron filter. Next, we looked at the proportion of samples that had at least one jumbo phage in them, which is on the x-axis here, at the different size filter size fractions, so the y-axis here. The dark teal are filter sizes that are above that 0.22 cutoff, and the light teal are below. And as you can see, jumbos could most frequently be found in samples above that 0.22 micron cutoff. Next, we looked at which filter sizes jumbos were most abundant. And so here on the x-axis is the relative abundance of jumbos in a sample. And on the y-axis are those filter fractions. Interestingly, jumbos were most abundant in samples from 0.1 to 0.22 microns on average. However, this 0.22 micron and everything below is the most commonly used filter, and it had the lowest abundance of jumbos in those samples. So this led us to conclude that commonly used filters in virology exclude jumbophages and reduce our ability to recover them from samples. Next, we wanted to ask, how do these jumbos matter? And we looked at how they affect the ocean's nutrient cycles in a few ways. First, we looked at where they are found based on which metagenome samples they were found in from that Terra Oceans data set. And then we looked at where they are most active, and we used metatranscriptome data for this. Metatranscriptomes is a fancy word for the gene expression data of all organisms in a sample. So the higher the gene expression of an organism, the more active we proxied that organism to be. And then we looked at what bacteria these jumbos might be infecting based on what genes they have. So to start, we found that jumbos are most abundant in coastal environments. So here's a map of all the samples we looked at. The size of the dot is its relative abundance. So the bigger the dot, the more abundant the jumbos are in that sample. And the dots are colored by their biome, which the 4 biomes are listed over here in the box plot. And this is a box plot of the abundance of jumbos in each sample separated by their biome or environment. And as you can see, the jumbos were most abundant in coastal samples. And similarly, we saw that jumbos were most active in coastal environments. So again, the bigger the dot, the higher the level of gene expression of jumbos in that sample, and the more active we inferred that the jumbos were to be in that environment or that sample. So the fact that jumbos were most abundant and active in coastal environments made sense to us because jumbos can only infect bacteria that are bigger than them. And in order for a bacteria to be big, it needs a lot of nutrients. And nutrients tend to be highest in coastal environments. So these results made sense. Next, we looked at what bacteria they were infecting. And we found that most jumbos were infecting proteobacteria, and a lot of consumer bacteria belong to this proteobacteria group. and very few were infecting cyanobacteria, which cyanobacteria can do photosynthesis in our algae. And this also made sense to us because these cyanobacteria tend to have smaller cell sizes, so it's less likely that a jumbo can infect them. And so we inferred that jumbos may be affecting the consumer microbial loop most in these ocean food webs. So in conclusion, or in summary, we found that jumbophages are widespread in the environment, but traditional methodologies have overlooked them. And we found that jumbos likely have their greatest impact in coastal environments where nutrient concentrations are high and available bacteria host cells are large. These 0.22 micron filter cutoffs are used to study viruses in all types of environments, so it's very likely that jumbophages are being overlooked in other systems. And they could be impacting the food webs and nutrient cycles in all these other systems like soils and guts and lakes and streams. So if you're interested in looking for jumbos in your system, please reach out to me. I'd love to hunt for phages with you. And with that, I'd like to thank my lab and the IGC and my funding sources and these kitties for keeping me in line during these work from home times. And with that, I'll leave you with a Backstreet Boys reference and take any questions. You can email me at elenarw@vt.edu. Thank you.